All right, so we finished our DNA. We know about our DNA structure. We know how to replicate our DNA. Now we need to use our DNA what it's for. We know that DNA makes our proteins. So this entire unit, protein synthesis, is going to take us through how we go from DNA to a protein. We call this the central dogma of biology, or molecular biology, which is just a fancy way of like our main principle, the main goal of our biology. If I All right. So what we're going to be doing is making our proteins. So a reminder of our proteins, this was one of our major macromolecules that we talked about a couple weeks ago, and they make up everything about us. Every chemical reaction, every enzyme has a protein. Proteins have to do with our hormones, so communication through our bodies, our appearance and our structures. A lot of those are proteins. So they are essential to our life. They are essential to who we are. And they are very, very, very important, which is obvious since our DNA makes proteins. So if our DNA is as important as it is, and the main thing it does is make proteins, that tells you just how important these proteins are. Your body right now is making thousands of proteins in your cells using your ribosomes. So remember, our ribosomes are our organelles that are going to make our proteins. Are ribosomes in both prokaryotes and eukaryotes? Do you remember? Yes, this was the one structure that is found in both prokaryotes and eukaryotes. So bacteria and all living things. So every living thing that has DNA, which is all living things, has to have a ribosome, because there's no point in having DNA if you can't turn it into a protein. You're going to go to joinpd.com. LBQUYM. All right, so your notes are for you. The Pear Deck is what's going to pop up questions. So that's how you get credit for. You don't need them if you don't want them, but I suggest doing them. All right, so protein synthesis is just a really fancy way for us to say making proteins. Biology, anytime it uses the word synthesis, it's just that's making. That's literally just a fancy way for saying making. So we're going to be making our proteins at a ribosome, and we're going to do it in 2 stages. So first is transcription. This is going to happen in the nucleus, where we're going to turn our DNA into RNA. And then translation, which will be at our ribosome, where we'll take our RNA and turn it into proteins. All right, so here's your first question. What is the function of a ribosome? All right, so we're making proteins. All right, so this is an RNA. This is our other type of nucleic acid. So this is ribonucleic acid. It's very similar to DNA, but has a few different types of quirks about it. The first is that it is single-stranded. So here's our DNA. It has 2 strands. It's twisted in that double helix. My RNA is just 1 strand, no particular shape to it. It's just a single strand of nucleotides. DNA has 2 strands. So remember we said it was like a zipper? RNA doesn't even really spiral into a helix. It just stays out as a strand. It's just kind of like a thread. There's no particular shape. For this picture this picture's not even really a helix. It's just waves. It's just wavy. A helix would be a spiral, which this isn't a spiral. It's just a wave. because it's a 3D picture, and if it was a spiral, these little stabs, the little nucleotides would be spiraling around, but they're not. They're all on one side. All right, so our RNA also has different nucleotides. It does not have thymine. So our apples in the trees is now changing. It now has uracil. So now uracil goes to adenine. The way I remember it, where do you tend to find apples that have fallen? Under the tree. So apples under the tree. A's either go to U's, under, if it is RNA, or T's, thymine, if it is DNA. This is the only time your nucleotides change. Cars are always in the garage. The cytosine always goes to guanine. But when we are talking about our RNA, our adenine, our apples are going to be under. It's going to go to your uracil. This tends to be something that catches people up a lot on this test because this next test is making sure you are reading. If it says take this to mRNA, you use uracil. If it says take this to DNA, you use thymine. So it's all making sure that you are reading. We're going to practice it a ton, so don't worry. There are three main types of RNA. The first is mRNA. The M stands for messenger. It's literally going to take the message from the nucleus out to the ribosome, so it literally acts as a messenger. tRNA, it's going to transfer, and this is going to bring us our amino acids. So it's going to transfer amino acids to the ribosome. And then our RNA is our ribosomal RNA. So remember, we said bacteria do not have organelles, but we said bacteria have ribosomes. The thing here is that all of our other organelles have membranes. So they're kind of like tiny mini little cells. Ribosomes are actually made up of RNA and are just a bunch of RNA kind of clumped together. It does not have a membrane, and so that's why our bacteria has a ribosome. All right. So what is our RNA and what's it being used for? This in particular, we're talking about mRNA, which it does not do a good job on the notes here. I even almost suggest putting an M in front of this in your notes. Because the RNA that we're talking about on this slide is talking about messenger RNA. So our RNA is going to act as the messenger of our DNA. We said our DNA is extremely important. And what happens when my DNA mutates? Remember what happened to your name. It turned to chaos. Sometimes it did nothing and we were fine, but sometimes your name just became a hogwash of letters. Sometimes it stopped too soon. It was chaotic. We do not want stuff to happen to our DNA. It can literally cause diseases. We talked about sickle cell anemia and we talked about cystic fibrosis. That is just 2 of thousands of disorders that can result from mutations in the DNA. So we never want the DNA to leave the nucleus. It's in this nice, happy little house. We don't want anything to happen to our DNA. Going out into the world, aka out into the cell, it could be destroyed, it could be mutated, things could happen. So we're going to leave it in our nice little capsule of the nucleus. However, we need the message and the code that is in DNA. We need it to take it out into the cell. And that's where our messenger RNA comes in. Our messenger RNA is basically us handwriting the code and then taking it to the cell. So what I suggest here is imagine my heritage is Italian. And so my grandmother could have this cookbook of recipes from her great-great-grandmother that are all from Italian history, and it has all of her special sauces and pastas that are all handwritten and insane. But let's say my sister asks my grandma and says, I want this sauce recipe. My grandmother's obviously not going to rip out the page. There's no way my grandma's going to hand over this giant family heirloom. So my grandmother would most likely say, I'm going to write it down and I'll give it to you. Because we don't care what happens to that piece of paper. We care what happens to the family heirloom. And this is not a true story. I do. I have Italian heritage. I am Italian, and my grandmother does have recipes, but they were just things that she remembers. So not quite too specific. No, she has recipes, but not in this fancy handwritten book. But this is very similar to our DNA. Our DNA is like our family heirloom. It's very important. We don't want it damaged, but we need those recipes. We need that code. My messenger RNA is the handwritten version. We don't care what happens in messenger RNA. We can make more of it, not a big deal. You'll also notice in my message with my analogy, my sister only asked for one recipe. She didn't ask for the entire book. mRNA is very similar. We're only going to do a couple genes, 1 or 2 genes. We don't copy the entire DNA every single time. I'm going to go in looking for that one thing that I need, that one thing that I want, make a copy of it, and then get out of there. And so that's how my messenger RNA lines up with my DNA. All right, so let's look at our protein synthesis. You'll remember seeing this image on our last lectures where we did our DNA replication. We talked that this was our central dogma, was this idea that our DNA will become RNA, and then our RNA will become proteins. What's the question? Okay. And you'll notice in this image, taking DNA and turning it into RNA is going to be transcription. So that's my step 1 of protein synthesis. And then you see that my RNA, we follow this arrow, becomes proteins. That's going to be my translation. So these are my 2 stages of protein synthesis. What we went over last unit was DNA becoming DNA, which is your replication. And you'll notice in today's graphic organizer, I've given you the protein synthesis version of these arrows because this idea is very important. All right. So, what is the goal of protein synthesis? Write your response. My goal of protein synthesis is to what? What am I wanting to make? Yep. Yep. Very good. So that's my stage 1. Very good. So I'm going to be making my mRNA to take outside of the nucleus, and then my mRNA is going to make what? It's literally the name of the lecture. Proteins. Our goal is to make proteins. So we're going to make our mRNA, and then our mRNA is going to go and make our proteins. All right, so let's look at step 1. This is transcription. So this is going to happen inside the nucleus where my DNA is. And I am going to be making my mRNA. So we're going to be looking for a particular gene. So let's say I just drank a bunch of milk. I need lactase, which is the enzyme that breaks up the sugar in milk. I'm going to go to my DNA and I'm going to find that one gene. And that's where I'm going to start copying. And I'm going to write my message in the form of messenger RNA, mRNA. And this process is very similar to our DNA replication. Our DNA is going to unzip, and we're going to match up nucleotides with the code that we want. But you'll notice he's only doing it on one side because how many strands does RNA have? 1. So he's only making 1 strand because that's all I need to have my message. What is the other different thing that's going to be with my RNA? What else am I going to be missing? As I'm making my code, will my A's be going to use? So my ATGCs are now going to be AUGCs. All right. So with that in mind, this is your code of DNA. I want you to turn this DNA into messenger RNA. What will I not have in my answer? T's. I'm not going to have any T's in my answer. All right, so my first letter, my A, what's my A going to become? A U, because I do not have T's. What's my C? G. What's my T? A. I still have A's. I heard a U. I still have A's. So my T's still become A's. It's just my A's do not become T's. All right. What's my G? C. What's my C? G. C. A. U. Perfect. So you should have U, G, A, C, G, C, A, U. And you yourself just transcribed inside the nucleus, and you have undergone stage 1 of protein synthesis. All right. Now we are ready to leave the nucleus. We have our message. We have our messenger RNA. We leave the nucleus with our mRNA, and we go and we find a ribosome. Our ribosome is going to read our secret code. It's going to read these nucleotides, and it's going to make it into a protein. Now, if you remember, when we were doing our DNA alias, how many nucleotides at a time did we read? How many nucleotides were one letter? How many nucleotides were in one letter in our DNA alias worksheet? 3. Remember when you were looking at your chart, you were looking for CAGs, TAGs, GCTs. That is because that's how DNA is read. A codon, which is what we called it in our DNA alias, a codon is just 3 nucleotides. And our ribosome is going to read 3 at a time, and it's going to pair up these 3 words with an amino acid. Remember, proteins are made of amino acids. So my DNA is going to be putting almost beads on a string to make my protein. And the 3 nucleotides, or that 1 codon, is going to be comparable to 1 amino acid. So just like in your worksheet, 3 letters equaled 1 letter. Here, 3 letters equals 1 amino acid. And we call these codons. So what's going to happen is our messenger RNA is going to squeeze right here. So this is a ribosome. It has a large and a small unit. And our messenger RNA is going to squeeze in between the 2 of them, and it's going to start reading the message. All right, here is our second type of RNA. This is our transfer RNA. I am aware that this part's going to be a little bit confusing. Transcription, pretty easy. Translation is a little convoluted, which is why we're going to go over it again tomorrow. That's why we'll go over it again Monday. We'll go over translation a few times because it does get a little bit complicated. So translation is when I'm reading my DNA and I'm turning it into a protein. We're not in math. The way to think of it is translating from one language to a next. I'm translating from the DNA language, the nucleotide language, and we're translating it into protein language. So that's what we're doing here. We're translating from one language to the next. All right, so I need to somehow get the correct amino acids to their location. And that is where my tRNA comes in. It's called tRNA because it looks kind of like an upside down T. So this is my tRNA. And at the very tip of my tRNA is called an anticodon. So in my mRNA, I have a codon, which is 3 letters. The anticodon is the reverse of those 3 letters. So if you can see here, my mRNA is AGU. The anticodon is the opposite of this, UCA. And they literally pair up like magnets. So if my mRNA comes in as AGU, it is always going to attract the tRNA that has the UCA anticodon. If they don't match up perfectly, then they do not pair together. Does that kind of make sense? Kind of. All right, and so you'll notice at the top of my tRNA is my amino acid. So my codon matches up to my anticodon, and it brings the proper amino acid, makes my protein, drops the amino acid off, and it goes and it leaves. So I'm going to pop up onto this picture. I know that's coming. Oh, you're not going to play my picture? All right, hold on. I'm going to pull this up then because the picture kind of puts it into, whoa. There it goes. So here's my ribosome. He's walking along. This is my messenger RNA. He's the one that brought me my message. And you can see my tRNAs are coming out of nowhere, but they have an amino acid. So they're grabbing their amino acid. They're matching up with the proper codon. They drop off the amino acid and they leave. This is a very simplified version of what is going on. So my ribosome is just moving my mRNA, keeping that message moving. My tRNAs are pairing up their anticodon with the codon of the DNA, and they're dropping off that amino acid and leaving. And so that growing pearl necklace that you see, or the string of beads that you see growing off of the screen, it's a protein. Well, it is getting bigger just off the screen. Sorry, Edpuzzle will show all of it to you, the giant whole protein. But so this is what's happening. They're matching up their codon, their anticodon, and they are making that protein. And so we are literally going from nucleotides to amino acids. That's that translation. All right, let's go back to, where's my pear deck go? Here it is. Okay. And so there's a little bit more complicated, more sciencey version of what we just saw. So as our ribosome is reading the codon, it's reading my 3 nucleotides, it sends out a signal saying, hey, tRNA, we need the tRNA with this anticodon. And the little happy tRNA comes along, drops off its amino acid, and it leaves. And so we then have that growing protein off to the side. Our amino acids are going to bond together with these things called peptide bonds. So I keep referring to my protein as beads on a string. The beads are the amino acids. The string is that peptide bond that's holding all of them together. And so once it has dropped the amino acid off, the tRNA is going to leave empty-handed and go and find a new amino acid so that it can be used again. So our tRNA is transfer RNA because it's transferring amino acids to the proteins. Kind of makes some sense. As long as it's a little bit making sense to you right now, like, okay, I kind of ish get it, we'll get it really into your head over the next couple of days. Here's our happy picture and our codon chart. So you need to be able to take your messenger RNA, turn it into transfer RNA, and then turn that into codons. I'm going to leave this one for another day, though, because I don't want to completely mind boggle you today. So we'll get this part done later on. So we'll do codons. We'll start codons later on. We'll start codons later. All right, so